Next slide. Okay, let's start with that of the probiotics or the live bacteria and the yeast, which are good for health and are beneficial, has effect on the host by improvising this intestinal and microbial balance. Coming to the history of probiotics, is just a part of the glimpse of probiotics I just want to share. At the start of 20th century, Russian Nobel Prize winner and father of modern immunology, Eli Metchnikoff, a scientist at the Pasteur Institute, was the first conceptualist who has defined the term called probiotics. Next slide. Next slide, please. Can you move on to the next slide, please? Okay. Just we go on to the characteristics of the effective probiotics. They are able to survive the passage through the digestive system, which are able to attach to the intestinal epithelia and brings about the colonization of good bacteria. And it is able to maintain a good viability, and it also produces to bring about the utilization of nutrients and substrates which has been acquired from the normal diet. This is the characteristic effect of the use or the taking of the probiotics in our dietary plan. Next slide. Non-pathogenic, and it is the nature of this probiotic bacterial system or the yeast. They are all said to be non-pathogenic and non-toxic. That is why we call them as the beneficial agents, which are all capable of exerting beneficial effect on the host system, and it has the stability of desired characteristics during the processing, storage, and transportation of the metabolites. And they have the properties such as anti-inflammatory, anti-mutagenic, and immunostimulatory properties. Next slide, please. You can move on to the next slides. Okay, what's the advantages going for the probiotics? They in turn produces the very important essential components such as lactic acids, which is very much essential in order to lower the pH of the intestine, thereby reducing the acidity and inhibiting the bacterial systems, thereby villains such as the Clostridium, Salmonella, and Shigella, and E. coli. And they also decreases the production of a variety of toxic and carcinogenic metabolites, thereby reducing the pH. They are not able to produce that of the deleterious effect. Besides this, it aids in the absorption of minerals, vital minerals, especially calcium, due to the increased intestinal acidity. These are some of the sources of the probiotic foods. Some fermented products containing similar lactic acid bacteria, which include pickled vegetables, fermented bean, such as the temp, hef, and etc. Besides this, buttermilk, kimchi, and soy sauce, which are all profound with the probiotic source of foods. Next slide, please. Okay, coming to the specificity of the medicinal plants, now I'm going to relate to the importance of the nutribiotics to that of the medicinal plants, because my specialization, which confines to that of the plant therapeutics and the medicinal plants, in this specifically, I'm going to focus on to that of the particular class of the fruit called as the Physalis minima. Coming to that of the medicinal plants, which are the rich source of secondary metabolites and as a potential source of drugs. These secondary metabolites, which includes alkaloids, glycosides, coumarins, flavonoids, steroids, tannins, etc. Medicinal plants has curative properties due to the presence of various complex chemical substances with the diuretics, antibacterial, antioxidant, and also the anti-cancer qualities, which can be found in that of the plant sources. These plants, which are acts as a natural antioxidants, they are successfully combat free radicals, averting oxidative damage to a vital cellular constituents. Herbal medicine is one of the oldest forms of the medicinal treatment in human history and could be considered as one of the far more process of the modern pharmaceutical trade. Physalis minima, this is the source which have been selected to in order to signify that in this particular plant source, namely the fruit called as the Physalis minima, which is enriched with that of the so much nutritional as well as that of the antioxidant property. This Physalis minima, it belongs to that of the family kingdom called the Tracheidae, and the family is Solanaceae, genus is Physalis, species belongs to that of the Physalis minima, and it has got an enriched source of vitamin C, and which is the importance of this vitamin C as a nutritional concept. It controls the blood pressure and it serves as anti-diabetic, and it has got wide ethnomedicinal use, and namely that of the blood purifier, and cures the nervous disorders, improves the memory, heals the wounds, and it also has find an application in the curation of the kidney disorders. And this plant contains, it has got enormous medicinal properties, namely diuretic. It serves as a diuretic agent, purgative, analgesic, antihelminthic, and also vermicelli killing and anti-inflammatory and antipyretic potentials. Next slide. So this plant was just been identified from the wild source, and it has been properly authenticated by the authentication process instituted by the of the Madras University. Next, next slide, please. Okay, this is an overview of the phytocompounds present in Physalis minima. It is enriched with that of the different types of the secondary compounds, namely polyphenols, vitamin C, and it contains, otherwise you can call it as tocopherols, and it contains set of flavonoids, terpenoids, alkaloids, and glycosides. These are the phytoconstituents which we are going, which are completely we have isolated, and we have been working on in this particular aspect. Next. Okay, this is the protocol what we have deduced for this particular talk. We are just going to make it out for the genetic variant analysis and screen for the phytochemicals, and also to study the antioxidant potential of this Physalis minima fruit. First, we start with that of the genetic variant analysis. Next slide, please. Okay. Just to give a glimpse of the genetic variability, it is a measure of tendency of individual genotype in the population to vary from one to another. Why we have to go for this particular process of genetic variation? Because variability is an important factor in evolution, as it affects an individual's response to environmental stress and thus can lead to differential survival of organisms within a population due to natural selection of the fit variants. So that's why we are just going to check out for the variation in that of the different sources of the plant what we have collected. Next slide, please. Okay, what may be the causes for that of the genetic variants in the natural process of the same type of plant varying to different environment? The reasons may be due to that of homologous recombination, or emigration or translocation, or due to polyploidy or diffuse centromeres or genetic mutations. These are the causes for the chance of the any natural selection system getting varied. Physalis minima plant, we have done a genetic variant analysis, and this plant source has been collected from 4 different zones of Tamil Nadu, that is east, west, north, south. So based on that, this first source is from that of Satyamangalam Kolli Road, and second one from that of the Kolli Hills, it's a wild fauna, where we call it as Namakkal. The next one from that of the Salem, and another one from that of the Dharmapuri district. So these are the different zones. From the picture, you can see the variants of the same plant that getting altered due to that of the environmental or different process phenotypic changes in the system. Next slide, please. What we are just coming out to that of the different sources of the plant variants has been taken with that of a technique called RAPD analysis, PCR-based method, which employs a single primer of arbitrary nucleotide sequence with the 10 nucleotides to amplify the PCR fragments from the genomic template DNA. So this analysis provides the change in that of the variants, which will be given on in the polymorphic difference that we'll see on to that of the results. Next slide, please. Okay, this is the outcome of the results what we have been talking about, the genetic variants of the Physalis minima, which we went on to that of the RAPD analysis. So you can see from the figure, lane 1, 2, 3, 4, 5, 6, this all just gives that of the different types of the bands which has been separated as per the variant. Previous slide, please. Can you move on to the previous slide? Okay, thank you. So this is the look variance, and these are the different areas from which we have collected the sample sources, namely Dharmaburi, Kolli Hills, Satyamangalam, and from Salem. Then they varying on the different zones and the altitudes and effects. The polymorphic band also varies accordingly. From Dharmaburi, we received that are six polymorphic, whereas five monomorphic bands. In case of that of the location selected for Holly Hills Normacal, we got 5 bands in polymorphic and there's no change in also monomorphic. Whereas in case of lane 4, that is here, we have this Physalis minima from Satyamangalam A Road, which provided the 7 polymorphic bands, whereas the monomorphic seems to be 5. And similarly, in case of Physalis minima, which is taken from that of Salem, which is a terrestrial process area, where we acquired and polymorphic bands 6 and monomorphics to be 7. So this reveals that the lane 4, what the source we have selected from that of the Satyamangalam, which holds a good polymorphic variation when compared to the rest of the other 3 sources what we have collected. Next slide, please. So the results of the 6 polymorphic bands, and which has been from that of the Dharmaburi district, 5 from that of the Namakkal, and 7 from Erode, and 6 from that of the Salem. So from this, we conclude that Physalis minima among the 4 districts successfully distinguished with that of the RAPD analysis, which makes us to quickly, and it's a simple tool for the identification of the plant, and also it provides the conservation and sustainability use of the phenotypic process. Next. Next, coming to that of the phytochemical screening of the secondary metabolites present in Physalis minima fruits, we initiated with that of the thin layer chromatography from that of the fruit extract, and this gives on to the results which gives the positive yellow spot, which indicates the presence of the secondary compounds, namely tannins, and next one is said to be a flavonoid, and third is said to be phenol. So these are the 3 prime secondary metabolites we have been screened with that of the thin layer chromatography from the Physalis minima fruits. Next. Followed from that of the confirmation with the qualitative analysis, and these are the general protocols we used to follow to go for the screening of the phytochemical constituents. We call it as the phytochemical qualitative screening with the standard procedures. Next slide, please. Okay, this is the result outcome of the phytochemical analysis of the Physalis fruit minima fruit extract, and there are it contains a wide range of the phytochemicals, namely alkaloids, glycosides, flavonoids, phenols, guanines, saponins, steroids, tannins, and terpenoids. Among this, we specifically this depicts the moderate presence, whereas that of the this tool, namely called as the single positive presence, which gives the moderate, whereas the higher level is denoted to be that of the double plus. Next. Next slide, please. Followed by that of the qualitative and TLC check, we have went on to go for the quantification of the phytochemicals present in the Physalis minima fruit, and we have went on to see that of the estimation of the flavonoids, phenols, tannins, and steroids, which has been confirmed with the TLC. Among the different types of the varying type of the secondary metabolites, namely flavonoids, phenols, and tannin steroid, this fruit upholds the higher content of flavonoids, followed by that of the phenols, then that is of the steroids, and finally tannins. Next slide, please. So after getting on to that of the wide spectrum of the 4 different phytochemical constituents, we wanted to just look out for the chemical profound content of this Physalis minima fruit. So we just went on to see with that of the characterization using GCMS analysis, and in this particular analysis, with the outcome of the result specifies that it contains 18 different phytoconstituents present in the Physalis minima fruits. Next. And these are the different types of the compounds which has been retrieved from this particular source directly. 18 compounds has been screened. Next. Next slide, please. Okay. Followed by that of the phytoconstituents. Next, we wanted to find out the chemical composition of this particular secondary metabolites present in the source. So this has been subjected to FTIR analysis, that is in order to find out the various functional groups which has been present in the particular source. So this graph or the picture gives the entire spectrum of the FTIR analysis of the secondary metabolites, which contains the functional groups which is essential for the therapeutic application. Next. So next, followed by that of the characterization, these are the different types of the compounds which has been identified with that of the varying chemical constituents, namely alkyne, amine, aromatic compounds. It contains the amine groups, and also besides that hydroxyl groups and aldehydes, alkane, which all will be later on utilized for the structural characterization of this particular phytocompounds, which will be later on purified and isolated. Next slide, please. Previous slide. Previous slides. Okay. So the second phase of our study is focused on to that of the antioxidant potential of the Physalis minima in the ethanolic fruit extract. This is a summary of the entire work which has been explained later. Move on to the next slide, please. So before going into antioxidant, it's just initial glimpses of the free radicals. Free radicals are the atoms, molecules, or ions, which are unpaired electrons in the outer shell configuration. These free radicals may have a positive or negative or no charge. Even though they are unpaired electrons by convention, metal and their ions or complexes with the unpaired electrons are not radicals. Unpaired electrons causes radicals to be highly reactive. Next slide, please. So radicals are the atoms, molecules are ions with unpaired electrons, and they are out of the cells. You can move on to the next slide, please. Okay. So in order to counter these free radicals, we need of the scavengers. They are called as the free radical scavengers. What are free radical scavengers? They are the substance which also known as the antioxidants, which helps to protect the cells from the damage caused by these free radicals. Free radicals are unstable molecules that are made up of during the normal cell metabolism. So the unpaired electrons can be counteracted by the radical scavengers, which are nothing but our antioxidants. Next. So antioxidants are the chemicals that can prevent or even slow their cell damage. Naturally, antioxidants are mainly found in fruits, vegetables, and in marine plants, and some seafood that eat the marine plants. Antioxidants can also be produced artificially and consumed in supplement form also. So Physalis minima fruit contains this antioxidant. We are just going on the assessment of the antioxidant activity in the ethanolic extract of the Physalis minima fruit was carried out for providing its utility in free radical mediated disease, including diabetes, cardiovascular diseases, cancer, etc. The ethanolic extract was screened for the in vitro antioxidant activity in order to assess the hydroxy radical scavenging, DPPH scavenging, and glutathione reductase and catalase assays. These are the assays were been taken out in order to exemplify the antioxidant potential of Physalis minima. Next slide, please. Okay. Coming to that of the DPPH scavenging activity, then the ethanolic extract of this fruit extract against the ascorbic acid, it shows that it's a very good effective potential from 0.920 milligram per ml and 0.43 per milligram per ml in this. The fruit extract exhibited a significant dose-dependent inhibition of DPPH activity. The DPPH assay scavenging activity is nearer to that of the standard active ascorbic acid. So this blue color, which is going to be our standard, and followed will be this red color which has been indicated in the graph, which is going to the fruit extract. So when you see that of the effective antioxidant potential activity initiated by the Physalis minima, it is going closer to that of the standard ascorbic acid, which confirms its antioxidant potential. Next. Next is for hydrogen peroxide scavenging activity. Here also, the ethanolic extract of that Physalis minima against the standard ascorbic acid is compared, and the ethanolic fruit extract of Physalis minima showed the highest hydrogen peroxide scavenging activity. When compared to that of this is a standard, followed by the nearer to that of the standard. It's a good sign of the antioxidant property. The next is the catalase, and the ethanolic fruit extract of showed an good antioxidant activity in against the catalase in scavenging that of the free radicals in this particular assay. Next. So glutathione reductase assay, and the reducing power of the ethanolic extract of Physalis minima increases with that of the increase in concert, which indicates the dose dependent. As the concentration increases, that is the activity also goes as such. So that needs to be standardized. This is a positive indication of the antioxidant potential of Physalis minima against the glutathione reductase activity. Next slide, please. So followed by this is said to be the non-enzymatic antioxidant activity, which we have been quantified from this Physalis minima food. The non-enzymatic antioxidants, nothing but vitamins, they play an important role in the biological defense mechanism of plants. Namely, they are ascorbic acid, and it's only very essential for maintenance of the antioxidant property. It prevents the plant from oxidative damage. The formation of the reactive oxidant species, which has been releases to that of the free radical, is prevented by these non-enzymatic antioxidant systems. Vitamin A, C, and E. Can you go on to the next back slide? Previous slide, please. So in this specifically the quantitative analysis, which reveals that the Physalis minima contains a very high concentration or good quantity amount of the vitamin C compared to that of the vitamin A and vitamin E. So in this case, it's going to have a very essential antioxidant property with that of the vitamin C. Now, this is just our entire plant therapeutic labs on which we are working on to that of the herbal medicines and also isolation purification. In our lab, we have the specificity of going for that of what is the importance or visionary to establish this lab. And this exactly gives the all natural sources. As probiotics emphasizes on that of the natural sources has got a very important probiotic effect and nutri effect. So our lab is focusing on working with that of the natural products and developing that of the herbal medicines based on that of the natural sources. So the entire process of a laboratory focuses on the visionary, where we say the nature has been always stands as a gold standard, marks and exemplify the outstanding phenomena of symbiosis, who recommends and encourages the use of traditional, herbal and natural remedies in this healthcare sector due to that of the easy availability and affordability. Herbal medicine involves alleviation of the disease by plant-based formulation method. This is our vision of our laboratory. And in the mission, we wanted to just say that traditional insight embedded with that of indigenous system of medicine needs a scientific validation, especially in its chemical parameters. Next. So these are the nearly or 30 different plants and also all related sources on which our lab nearly working for 20 years on herbal medicines and also the on different clinical manifestation, namely liver disease, kidney, and anti-inflammatory, and we have been working on to that of the liver fibrosis also. So these are the different plant sources we have completely isolated and ordered and also now used as a therapeutic drug for different conditions. So I'm happy to share with all these views that they are all contain very important essential probiotic, not only probiotic and nutritional properties also in this indulged. Next slide, please. Almost all different parts of the plants we have been using entirely in our lab. So this is the protocols we used to follow for any source of the plant in our laboratory. From the natural source, we get it on, and we go on to that of the extraction with that of the different oxalate extraction protocols. Followed by that, we perform a qualitative, quantitative process and identify the compound, and then we characterize the compound identification with that of the GCMS, FTR, UV, and then SEMTEM, etc. Then followed by that, after getting on to the compound isolated and purified, we check out for that of the radical scavenging activity, that is in vitro radical scavenging activity, and followed by that, finally, we end up with that of the in vivo animal study, where we emphasize the toxicity limit and also specify the clinical manifestation to different conditions from that of the source. And then we perform the cell line studies on cancer cell lines and also normal cell lines to check out the effectiveness of the in vitro. And finally, we are going to go for the drug discovery with that of the in silico molecular studies, where we just express that of the compound what we have isolated and purified later on can be completely exhibited as an effective drug against that particular clinical condition. So this is the format of the protocols we used to follow in our lab. So this is our entire protocols we are following. Thank you for your patient listening. I'm happy to share the content of the Physalis minima has to be a very important essential probiotic. We can utilize to have a natural source which contains an enriched level of vitamin C, besides that all other essential nutripotentials. Thank you one and all for your patience. And any questions can be clarified. I want to come, just go to the last slide, please. If you don't mind. Okay. So this is the final message I want to say. Phytomedicine a day keeps all the ailments away. Wish you all a healthy, happy life. Thank you all.